Hello and welcome to a fifth annual Drug Discovery and Development virtual conference. My name is James McKim. I'm a biochemical and molecular toxicologist by training, and I've been working in the area of in vitro toxicology for about 25 years. In the last 20 years, we've been focused on the development and validation of new methodologies, and those methodologies have been supporting the replacement of animals. That's our brass ring, if you will, for the future. And we've been working in academia, chemical industry, and pharmaceuticals. And I'm looking forward to talking to you today about the work and the progress that we've made on in vitro platforms that allow integrating organs through simulated blood systems. And so I'm going to be talking to you about that platform that was developed here at IONTOX for understanding pharmacokinetic and toxicologic effects of drugs and chemicals. So why do we need an integrated organ platform in vitro? Well, it's very important that over the years, we've seen great improvements in both tissues and the analytical technologies for in vitro work for the testing of drugs and chemical safety. But now we need to understand relevant routes of exposure and what happens after the exposure organ and we get to the delivery organs, the systemic exposure. How do we model that in vitro? We want to be able to study the effects of multiple organs on the test chemical. We want to be able to look at the movement across multiple biological barriers. And we want to be able to develop pharmacokinetic data and estimate key parameters, for example, the AUC or half-life. And we want to be able to understand repeated dosing. So we want a repeat dosing dynamic model. And then most importantly, we want to be able to have the ability to extrapolate this in vitro data to in vivo reality. So multi-organ methods provide important data that you can't get from a single cell system. We can start to ask questions about absorption, distribution, metabolism, etc. And to do that, we need to be able to study key interactions. The three primary routes of exposure, the oral, dermal, and lung or respiratory. And therefore, we want to look at interactions like the intestine to the liver, skin to liver, lung to liver. And there's a lot of science and business advantages to these types of systems. We should be able to reduce time of new drugs and chemicals to market. We should be able to start predicting, without using animals, what's happening in humans. This would greatly reduce developmental costs, and it would rapidly move us towards really a completely different paradigm in the evaluation of drugs and chemical safety. So I'm going to be talking to you about a human dynamic multi-organ platform. We refer to it as HUDEM-UP. And why HUDEM-UP? Why do we need this? And why is this model different? Well, for several years now, we've all been reading about organ on a chip. And although this technology is really quite brilliant in terms of its engineering, there are some disadvantages in terms of the biology, which I'll go over. And remember, all in vitro systems, all platforms, nothing is 100%. So there's advantages and disadvantages to all platforms. But one of the problems is that when you miniaturize a platform, you can reduce tissue mass to a level where you just don't have enough to do a lot of the biochemical and molecular analyses that you need to do. Fluid volumes get very low. And as a result, it's very hard to do repeated dosing without extremely sensitive analytical equipment. So if you want to do, for example, a plasma concentration type time curve, you want to look at how drug changes over time, you're going to want to take a lot of samples, and that can be difficult. Only certain cells can be used in these models, and most of the time they've been tumor cell lines. And frankly, we've really gotten away from tumor cell lines in the last several years and moved more towards human normal tissues and three-dimensional tissues. A key issue has been the types of plastics used. Typically, the plastics that were used initially were extremely absorbent to the chemicals. So it's very hard to detect the materials at low doses. The fluid compartments couldn't be isolated, and really it hasn't been successful in terms of linking multiple organ chips together to get a mimicked, human body or more physiologically relevant system. So while there are many advantages in terms of specific organ mechanics and how those mechanics might impact drug movement, kinetics, or toxicology, the overall system has some disadvantages. So we started with the idea that we need to build the science first, and then worry and the biology, and we're not going to worry about manufacturing and selling a plate until we know that a plate is capable of answering some key questions. And so we really started with a larger plate design. So this footprint, this diagram represents the footprint of the Hudimop, and it's a standard microtiter plate, a microcell culture plate size, and the wells are about the size of a 6-well system. And when we use this type of a system, we can connect them by using a simulated blood flow system. And what makes this really unique is that the only places where these different compartments communicate with one another is via the simulated blood, and that's because inside each organ chamber, there's a semi-permeable membrane, which is picking up and releasing the parent compound metabolites and even small proteins. And we can change the surface area of this semi-permeable membrane to mimic different parts of the body and different types of blood flow. And so in this representation here, we see again the diagrammatic representation. Although it looks rigid, keep in mind that these can be moved around, and that should be clear in the center picture, where you can see the cups that sit inside the plate, and those cups can be moved in any configuration that you need. And the black supports are for using 3D tissues. And then the fluid is moved through the system by using a micro syringe pump, which is shown on the right. And this is just an example of a 3D human lung tissue. This human lung tissue is mucillar, and it's made by Epithelix. And it fits very nicely into these black supports that allow us to link those systems together. So some of the important features then of this new design plate platform are that we're dealing with a larger scale. It's an integrated organ platform on a scale that allows us to use larger volumes and larger amounts of tissue. It's compatible with most commercially available tissue models, which I'll show you in just a moment. The simulated blood flow is unique. It's the only way the organ chambers communicate with one another, and that's again through a semi-permeable membrane that's only inside each organ chamber. And inside that simulated blood flow system, we have basically a saline solution with human serum albumin. And of course, that can be adjusted to any components that you would want to use. So each organ and tissue can be cultured under optimal conditions because we're not exchanging media from one compartment to another. There's no net change in fluid, in fact, between the chambers. There's only a change in molecules that have an osmotic gradient. We can calculate kinetic data because of this. And our plastics are designed and coated so that we have very low absorption of drugs and chemicals. We've used the model with a set, like I said earlier, many different tissue types, which you're all familiar with, and I'm going to show some of those to you now. I've already mentioned mucillar. This is the lung model. It's made by Epithelix. We've used cardiomyocytes, the I-cell system. We use primary hepatocytes in standard monoculture or in sandwich culture. And we've used the epi-intestinal system that's made by Mattech Corporation, and we've had excellent luck with this for our oral models. So these are just examples of the different tissue types that can be used in the platform. And other examples would include spheroids, and of course any cell line that you happen to have experience with in your laboratory. Well, when we designed this, we of course wanted it to look at least in principle like the human body. And this is just to remind us that organs are connected by the blood system, obviously, and that's why the simulated blood flow is important, but also the sequence of connection of one organ to another is quite important. Another important component is the idea of dilution. And when you're putting chemicals directly on cells in primary culture, it's very difficult to account for changes in fluid compartments in a body. But in this type of an in vitro platform where we're integrating different organ systems and we're using simulated blood flow, we actually start to account for different volume changes and those dilution effects. And this was just a diagram to give you an idea that as a drug enters the intestine by oral intake, it's absorbed, it's diluted in the intestine, but also it's absorbed in the portal system and diluted again in the liver. And those volumes are shown here on the right, the stomach liver volumes, the total volume of 5,000. And so if you just had an arbitrary dosing, you can see that that dilution factor is quite large. And so this ability to model this type of dilution effect is really quite important. This is just to show you that the outside syringes can be a single syringe doing a flow through or multiple syringes. We can change the flow rates. We can have it flow through 100%, or we can have it loop back in to the system. So again, the Hutemop system that I'm going to be showing you data for next it's not organ on a chip. It's mesoscale. It's not a full body on a chip. It's an integrated organ platform that allows us to start asking questions about drug or chemical movement, metabolism, toxicology, and kinetics. So now I'm going to move into some proof of concept studies to show you how the system works. And let's start, as I mentioned earlier, it's important to be able to look at different routes of exposure, dermal, oral, and inhalation. And in the first examples, I'm going to be showing you some inhalation exposures, and then later on, I'll show you some intestinal or oral exposure as well. So again, we have a MatTek system for skin, we have the lung system by Muselair, and we have the intestinal system by MatTek Corporation. And this is a side view of a two-compartment plate system, and the left shows you the three-dimensional tissue grown on the insert. Material is dosed at the apical surface, it diffuses through, and that allows us to monitor the movement of parent, but also any metabolites that are formed. That material is then taken up in the basolateral chamber by the simulated blood flow system, again across that semi-permeable membrane, and it's delivered to the next compartment, in this case the liver, where again we can measure the delivery of the parent molecule as well as the changes in the metabolic profiles. And all of this does a great job of mimicking the three primary fluid compartments and biological barriers in the body: the intracellular fluid, the intravascular fluid, and the interstitial fluid. So before we start a plate study or before we start working, we've learned over the last 5 or 6 years that we really need to establish an understanding of the system every time. And that includes the movement, metabolism, toxicity of molecules. And in the examples I'm going to be showing you now, we're going to be looking at diclofenac, a very well-known drug. We're going to be using a three-compartment or three-organ system, and this system is interesting. It's just dermal exposure to primary human hepatocytes, to a different type of hepatocyte over here to look at effects of metabolites from liver 1 on liver 2. So we're going to define nonspecific binding in a null system or a system with no cells or tissues. We're going to look at the cytotoxicity range, so what's the maximum tolerated dose we can put on without killing our tissues. We want to confirm that the organs that we want to be producing metabolites can actually produce those metabolites. We need to develop the analytical system, in this case LC-MS-MS, and then finally we dose the system, the full system, all linked together and start collecting the data. Again, this is a dermal exposure, just showing you again the insert and the doses applied to the apical side of the skin. And this is the first step where we are just looking at toxicity or maximum dose, and then on the dermal side, we're using a MTT assay, and this is percent viability. And you can see not much change in viability over 48 hours. In the liver, we're using the primary hepatocytes, and here we see pretty significant toxicity as the doses go up, expressed both in micromolar and in the mass applied. And then at the bottom, this shows the development of the analytical process and a nice peak for diclofenac. Now, diclofenac has, as you probably well know, has several metabolites. The parent compound is shown at the top left. That is hydroxylated, the 4-hydroxy, so we get a hydroxylated diclofenac, and it can also be glucuronidated. So we wanted to be able to know if our system could pick up these metabolites over time. And so the red bars on the left are showing you the formation of the 4-hydroxy diclofenac, and you can see at the 3 doses, we're getting a nice increase over time, 1, 6, and 24 hours of the metabolite. Now it's going down at 300 because we saw, remember in the last slide, we saw toxicity. And also the same is true with the glucuronide. Again, we can see the glucuronide going up in a time-dependent manner, and as toxicity occurs at the higher dose, of course, it goes down. We also wanted to confirm that the material moved across the skin, so we wanted to look at dermal absorption. And so again, we can see here, this is showing you just using the dermal cup to look at the movement. You can see a nice increase over time, and that allows us to calculate permeability coefficients, and this is the beginning of the estimates of bioavailability. So now we've got most of the pre-study work completed. We understand nonspecific binding. We understand what doses we could administer to the tissues without causing detrimental effects. We've confirmed the production of the metabolites that we want to monitor. And we've developed our analytical method. So now we want to take that 3-compartment system that I showed you a couple of slides back, which consists of the skin and the liver 1 and liver 2, and we want to look at developing the kinetic curves that we need to look at any type of ADME data. So this is diclofenac dosed at 15 micrograms of mass on the apical surface of the skin. And this is time on the x-axis, and the amount of diclofenac recovered on the y-axis. And you can see this is a 2-dose exposure. So we dose at time 0, we collect samples, and you see it nicely plateau right around 24 hours. And at 24 hours, it receives a 2nd dose. And again, we get a rise, much like you would in vivo, just as you would expect. If we look at the other compartments now, the light yellow line represents the simulated blood, and you can see the material went up quite rapidly in the blood, as you would expect in vivo as well. And there's a slight delay as the material moves across and into the first liver, which is in black, and that's lower, has a lower amount of recovered material than the red, because we're looking at parent compound, and in the black, or the first liver, we have much higher rates of metabolism. Remember, these are the human primary hepatocytes, and the other cells are a cell line. And you can see that is reflective in the material being delivered. And here we have a different dose. So the first dose I showed you right here was at 15, and you can see it plateaus around 8. We increased the dose to 30. And you can see now we've really gone up again. We're about 16, so we're getting a nice dose response here that's proportional to the dose given. The same is true in the different organ compartments. And we're also seeing a nice time-dependent increase in the first liver compartment of the glucuronide metabolite. So again, these kinetic curves are all being generated in this plate system, and all of these data can be modeled. When we look at delivery of metabolites to the second liver compartment, you can see that the metabolites or diclofenac itself are causing a reduction in total glutathione. So that tells us it's getting there. We've measured it there, but it's also having a biochemical effect. So now let's look at some other compounds. We looked at diclofenac. Now I'd like to show you how we're moving towards trying to estimate some of these pharmacokinetic or ADME parameters. So we're going to look at acetaminophen and cyclohexamide. We don't have time to look at ritonavir today. But let's start with those. So here we have a 3-organ system. This time we're looking at oral absorption, so we're using a 3D human intestine model, human hepatocytes, and human kidney cells. And we're looking for ways to calculate AUC, Cmax, and time to max. Again, we're looking at acetaminophen, which has low toxicity relative to cyclohexamide, an antifungal protein synthesis inhibitor, which has much higher toxicity. Now, this graph it has a lot of information, but let's just start on the left. The bar graph on the left is just telling us that the intestinal tissue is healthy. This is over several different exposures of acetaminophen, and you can see that the viability remains quite high, and the one that's quite low was the positive control, which is Triton X. In the middle, we have the kinetic data. The upper graph shows the absorption across that intestinal barrier and the elimination. The lower graph is a blow-up of the other organ compartments, which are liver, kidney, and the simulated blood. In this case, we labeled it perfusate. Again, we see these nice lags. This is a single dose. The first example was a double dose. This is a single dose at time 0. And then lastly, we looked at some cytotoxicity at the end of the experiment. Now, this is Ldh leakage on the basolateral surface of the intestinal chamber. This is the apical surface, and this is normal because these cells will slough off. You can see this is a model of that 3D intestinal chamber. These cells will actually slough off from the crypt just as they do in vivo, and so that increases the LDH from the apical wash. So we're focusing on the basolateral side. Again, really low toxicity, below two-fold changes in LDH. Out on the kidney side, in the liver, there's almost no toxicity. On the kidney side, we see a little bit more, and acetaminophen does have a higher toxicity in the kidney. We also looked at genes associated with toxicity, and this is just a table of those to give you an example. We looked at Bax BCL2, NERF, glutathione pathways, some cytokines for inflammation, and then some key CYP enzymes. And this table at the top shows you the gene changes. So this was done with RT-PCR. The organs are on the left, the genes across the top, and anything in green is more than a two-fold induction. And for the acetaminophen, the intestine, the liver, and the kidney compartments, not much happening, as you'd expect for a molecule that's very safe. For cyclohexamide, we see a little more activity. Again, not much activity in the intestinal tissues. It's pretty robust. But in the center, we see a nice increase across the intestinal model, but a delay in the elimination phase. And this is, we see this with changes in log P as well as molecular size. And then down the bottom, again, we see nice uptake in the other organs. There is more toxicity at time points here in the intestine. You can see that multiple time points now are nearly at 5. Again, the two big bars are the apical side. In the liver, we see significant toxicity and just a little bit at the last time point in the kidney. And again, this is from 2 to 48 hours of measurements. So again, we're looking at this this model and trying to predict the Cmax and the time to Cmax, and actually these values are really quite similar to what we see in vivo, and that was true when we did the acetaminophen as well. That time to see the Cmax and the Cmax values themselves. So it's very encouraging data for us as we move forward with these models. And for cyclohexamide, now we're looking at the gene expression profiles. Again, anything in green is 2 or greater in terms of its fold change. We see a lot more green, meaning a lot more genes are impacted across multiple organs. And again, the bars reflect that here at the bottom. And the dashed line just represents that two-fold threshold that I've been talking about. And then lastly, I just want to introduce, we have partnered with Vitro to develop an in vitro way to expose the plate, the 3D lung tissue on the plate, to aerosols and vapors. And this is just an example of what that system looks like. So in summary, then, I've shown you data, a lot of data actually, on the Hudemop platform, which is a microphysiology system that is based on mesoscale formats, which enables multiple tissue and liquid sampling capabilities. We're showing you that it's possible to generate very nice kinetic data. That means that you can model the system. Multiple organ systems can be monitored. Different groups can be put together. I showed you primarily, a lung, a skin to liver, a lung to intestine to liver, but we can also do lung, and we're also now experimenting with blood-brain barrier and neuro brain deliveries. The testing paradigm allows faster development. We believe it will increase the way, it'll improve the way we design and approve drugs. And we're very excited about working with various companies around the world right now, even developing this and validating the system even more. And the current iteration of the plate, it looks like this. Again, these cups can be moved around. It's very flexible to run 2 compartments, 3 compartments, 4, 5 compartments. This is just a picture again to give you a little better view of what it looks like. And with that, I will close and thank you for attending the webinar series today. And I really look forward to your questions and to answering them via email. Thank you.